Have you ever thought about what keeps a cell, well, a cell? It's this incredible structure, the plasma membrane. But here's the thing, it's not just some passive bag holding everything in. Think of it more like a bustling city's border crossing, a smart, dynamic gatekeeper that's constantly deciding what gets in and what gets kicked out. It's the living edge of life itself. So here's how we're going to break it all down. First, we'll look at the basic foundation of this border wall. Then we'll meet the specialized molecules that regulate it and the protein workers who do all the heavy lifting. After that, we'll dive into how things actually get across, starting with the easy, no energy required methods. And finally, we'll wrap it all up. All right, let's start from the ground up. Every cell needs a boundary, an essential barrier. For the cell, that barrier is something called the phospholipid bilayer. Let's see how this thing is put together. Now this right here is the famous fluid mosaic model. And the two words to focus on are fluid and mosaic. This barrier isn't a rigid brick wall. Nope. It's more like a thick, oily sea of lipids that are constantly shifting and moving around. And floating in that sea, you've got this mosaic of different proteins, each one with a very specific job to do. So what makes up this fluid sea? It's mainly these 5 types of molecules called phospholipids. You can think of them as the individual bricks that build our cellular wall. They all have a similar blueprint, but tiny differences between them are actually super important for the membrane's different functions. Okay, this might look a little complicated, but the main idea here is actually really elegant and simple. Every single one of these molecules has 2 distinct parts. A head that is hydrophilic, meaning it loves water, and then these 2 tails that are hydrophobic, they hate water. This dual personality is the absolute key to how the entire membrane automatically assembles itself. And here's another really clever design feature. The membrane isn't the same on both sides. It's asymmetric. It's kind of like a boat's hull, where the outside is designed to handle the ocean and the inside is designed to deal with the cargo. You can see here that the types of phospholipids facing the outside world are different from the ones facing the cell's interior. This difference is vital for things like sending and receiving signals. So here's the payoff. Because all those fatty acid tails hate water, they all turn inward, hiding from the watery environment both inside and outside the cell. This creates this oily, greasy middle layer, the hydrophobic core. And that core is what makes the membrane such a great barrier. It naturally blocks almost anything that's water-soluble from just wandering through. Okay, now let's introduce a new character, cholesterol. Now, cholesterol doesn't actually build the wall, but it's like a crucial foreman on the job site, moderating the whole structure and making sure it has the right consistency. So here it is. You can see it has this bulky, rigid ring structure. Because of that shape, it can't really form a membrane all by itself. Instead, it wedges itself right in between the phospholipids, and its main job is to act like a buffer, regulating the membrane's fluidity. And this is where it gets really cool. It has this dual role that depends on temperature. When things get hot, cholesterol actually makes the membrane less fluid, kind of stiffening it up so it doesn't get too leaky. But when it gets cold, it does the opposite. It gets in the way of the phospholipids, preventing them from packing too tightly and freezing solid. It's basically the membrane's personal thermostat. All right, we've built the basic structure. Now it's time to bring in the real workforce of our cellular city, the proteins. The lipids provide the arena, but it's the proteins that perform almost all of the membrane's specific active jobs. Now, these protein workers come in 2 main flavors. You've got your integral proteins, which are really embedded in there, often spanning the entire membrane from one side to the other. And then you have peripheral proteins, which are more like temporary contractors, just loosely attached to the inner or outer surface. And what do? Well, they have at least 6 critical jobs that transform that passive barrier into a buzzing hub of activity. Let's quickly run through what they are. First, and this is a huge one, transport. These proteins are the bouncers and the shuttle drivers, creating specific channels and gates for molecules that can't get across the oily core on their own. Next up, receptors. These proteins have a specific shape that lets them bind to signal molecules, like hormones. When a signal docks, it's like a key turning in a lock, telling the cell to do something specific on the inside. Some of these proteins are also enzymes. They're basically little chemical reaction factories, with their active sites sticking out so they can get to work right there on the cell's surface. These guys are called glycoproteins, and they act like cellular ID badges. Those little carbohydrate chains you see are unique to each cell type, which is how our cells recognize each other. Super important for our immune system. Then we have attachment proteins. These are like the cell's anchors. They link the membrane to the cell's internal skeleton on one side and to the external framework on the other, giving the cell its shape and stability. And family, there are junction proteins. These are what literally stitch cells together to form tissues, creating tight seals or communication channels between neighbors. Okay, time for a quick pause and think. Out of those 6 jobs we just saw, which one is most like a doorbell for the cell? I'll give you a second. If you guess receptor proteins, you got it. They sense something on the outside and ring the bell to let the inside of the cell know a message has arrived. So, now that we know the lay of the land, the lipids, the cholesterol, the proteins, let's look at how things actually get across this border. And we'll start with what's called passive transport, basically all the ways to cross without needing to spend any energy. The simplest way is diffusion. It's just the natural tendency for stuff to spread out. Molecules will always move from where they're crowded to where they're less crowded. It's just physics. And for small, nonpolar things like oxygen and carbon dioxide, they can just diffuse straight through the membrane, no problem. But what about stuff that can't just slide through that oily core? Well, for them, there's facilitated diffusion. It's still passive, it's still moving from high to low concentration, but it needs help. That help comes from either a channel protein, which is like a simple tunnel, or a carrier protein, which actually grabs its passenger, changes shape, and carries it to the other side. Now let's zoom in on a very special type of diffusion. It's so important that it gets its own name, osmosis. This is all about the movement of water, and it's absolutely critical for life as we know it. Here's the simple rule. Water always follows the solutes. In other words, water will always move across a membrane from a less concentrated solution to a more concentrated one, almost like it's trying to dilute the saltier side. And this simple rule has some really dramatic consequences for cells. For example, if you put a cell into hypotonic solution, like pure water, the inside of the cell is saltier than the outside. So what happens? Water rushes into the cell, causing it to swell up like a balloon. And for an animal cell without a rigid wall, it can actually pop. Now, the sweet spot is an isotonic solution. Here, the concentration of stuff inside and outside the cell is perfectly balanced. Water flows in and out at the same rate, so the cell is happy and stable. This is the environment our body tries to maintain for our cells. But what if you put a cell in a really salty hypertonic solution? Well, now the outside is more concentrated than the inside, so water rushes out of the cell, causing it to shrink and shrivel up like a raisin. Okay, another quick thought experiment. Why do you think it's so dangerous to get an IV of pure distilled water? Based on what we just saw, what would happen to all of your red blood cells? That's right, they'd suddenly find themselves in a seriously hypotonic solution. Water would flood into them, and they would swell up and burst. Not good. All right, let's bring it all home. We've seen the lipids, the cholesterol, the proteins, and the rules of transport. Let's do a quick recap of how they all come together to make this incredible living border. So, to sum it all up, we've seen that the membrane is a fluid mosaic, not a static wall. Its two-sided asymmetric nature, along with the stabilizing effects of cholesterol, is key to its function. The real action happens thanks to the proteins, which handle everything from transport to communication. And finally, passive transport is the simple, energy-free way that many molecules flow across this border, always moving down their concentration gradient. So we've established that the membrane is a barrier, sure, but it's so much more than that. It is the cell's primary communicator. Which leaves us with a final, pretty important question. If this elegant communication system is what keeps a cell healthy and functioning properly, what happens to us when those signals get crossed and that communication breaks down?